Hello again. Welcome to the second chapter about scientific software best practices. I'm Candace Savanen, your primary instructor for this course. Here's licensing information. All right, in this chapter, we're going to demonstrate how to recognize that development of scientific analyses should be thought of a form of software development. We're going to understand that automation is one tool of many that can enhance your software development process. We're also going to demonstrate how to advocate for environments that are more conducive to sustainable scientific analysis development. So, in theory, the scientific process goes like this. You have a question, you have some data that you either collect or someone else has collected, or you apply to a question, and then you find something and you publish it. When we summarize it like this, it all sounds very simple and straightforward and should be pretty quick. But, of course, it's not. Of course, really what happens in science is we find some things, we might have to resort back and ask some more questions, collect more data. We find some side questions and other investigations we need to go through. And it really ends up being a pretty complicated process with a lot of tangents and weaves and returning back to things. And so this can make the actual record keeping and the data analysis and the software that we develop, it can make this whole workflow a lot more complicated if we aren't careful. So the role of software in science is usually also then not linear either, right? We don't just collect our data, write some code, and then publish a manuscript all in one swoop. Usually, again, it requires a lot of additional side quests. This also means that complex analyses need to scale with more complex infrastructure. So what do we mean by that? So, for example, there in this hypothetical graph, we can think of these different regions. If you have a simple script, there's not too many different decisions in there. It might be easy enough to kind of share that with somebody and they understand kind of exactly what you did, and you don't really need a lot of complicated infrastructure for it. However, the more complex your pipelines are, the more decisions you made along the way, along that analysis, the more that we might need some more infrastructure to support that. We can also picture these other types of scenarios, right? In region 1, we might see an area which we have over-engineered a bit, maybe prematurely optimized, and then ended up not really using that. That happens sometimes, but honestly not very often because a lot of academic researchers are super busy and don't have time for this kind of stuff. The more common scenario that I think this course and others is trying to address is a scenario in which the analyses are very complex. So you see region 2 over there. We have a very complex analysis. However, the infrastructure to support that analysis may not have increased in complexity to support it. So this means that analysis that's very complex might not be very easy to reproduce, maintain, or modify. And this is really the very common scenario right now in research, and that there's tools like automation that can help mitigate this problem. And that's what this course and others are about. So let's go through some actual tangible examples. So a more simple analysis might be we have a t-test, right? If we said that we did a t-test in R or in Python, there might be pretty straightforward. There's pretty widely used functions for that. We still might want to make sure that we're describing like what kind of t-test we did and did we do multiple testing correction. But if we really only did one t-test, there was not too many decisions that we need to describe as compared to some other kinds of more complex analyses. So in another example, a more complex analysis is for sequencing analysis. So for sequencing analysis, there can be all kinds of decisions we made along the way, right? How did we sequence? First of all, what are the samples that got sequenced? How did we sequence those samples? How did we map those samples? Did we map those samples? What kind of parameters did we use to map those samples? What did we do for normalization? Did we normalize the data? If so, how did we normalize the data? With what methods? What parameters? How did we treat the controls in the experimental groups? How did we calculate the log-fold change? Like, there are so many decisions made along the way with any type of sequencing analysis, and especially the more complex sequencing analysis, the newer the technology and the innovation, the more, again, we're going to need to make it abundantly clear what decisions we made along the way. So this kind of example would require a lot more complex of an infrastructure that we want to be more robust if we're going to make sure that it's just as reproducible as our simple t-test. Again, just to be clear, a pipeline is usually the word we use to describe multiple scripts or programs put together to accomplish a whole set of tasks for a goal. So we can think about this then if we used automation, right? So without automation, we might have like 6 steps or something like that, right? Then that's a lot. That could be a lot if we're ever asked to reproduce this, which we probably will be. And so changes to the code might result in us having to redo all 6 steps of these. Now with automation though, we might reduce this to, maybe 2 steps, maybe less steps. And that is invaluable as a researcher. Not only because we're strapped for time sometimes, but also because there are a lot of things that robots are much better at than humans. Robots, if you tell them, hey, do this exactly the same way with these very explicit instructions, robots are very good at being repetitive. Humans, we have a little more variability. We're a little more creative. We're a little more unpredictable. And that's good for some parts of science. But for the parts of science where we just need the thing to rerun, we might want to consider using automation instead of human hands to do that. So in this course then, we're going to talk about how to automate because it is one skill set of many that helps promote reproducibility in analyses, whether they be simple or complex or anywhere in between. Reproducibility is aided by automation. So it's a really handy skill set to have in your back pocket. So excited to dive in more.